In our learning unit here on understanding how enzymes work, we are really going to have 4 enzymes that we'll study as sort of exemplars of understanding how this process works. 3 out of those 4 enzymes are going to be enzymes that catalyze the hydrolysis of polymers that we've learned about: carbohydrates and glycosidic bonds, peptides and peptide bonds, and then RNA and a phosphodiester bond. Okay, so the first part here is just going to be walking through and reviewing what we remember about polymer hydrolysis. What are the polymers that we know? What are the enzymes that we're going to study? And kind of what are the themes that we are going to see? And then we'll also sort of see this overarching theme of how we will understand that amino acids are able to help do this chemistry. And importantly, what are kinds of chemistries that amino acids can help with, and what are things that they cannot help with, and how do we have then assistance in that regard? So when we think about hydrolysis reactions, that's going to be a word that we'll see a lot. We're really talking about water doing breaking apart. So lysis is breaking apart by water. Okay, so really we're talking about taking these polymers, and these polymers have certain linkages, and I just reviewed what they were for you, but we'll go ahead and we'll write them down here. So these polymers have certain linkages that are going to be broken apart by the addition of water into their monomers. So why is this important? Well, many of these enzymes are ones that we're going to find in our digestive tract, and that's really important because only monomers exit your GI tract. So it's really important that you have these enzymes working properly to break those polymers down into their monomer forms to absorb them out of your GI tract. Okay, and so we have only monomers exit the GI tract. So only monomers exit our GI tract. Okay, so let's do a little review here about the 3 polymers that we're going to talk about. We've got carbohydrates, we've got nucleic acids, which really are DNA, but we're focused on RNA hydrolysis here. And then we're going to have proteins. So those are the 3 polymers that we're going to talk about. I said that there were 4 enzymes that we're studying. We're actually going to study the enzyme chemistry of carbonic anhydrase. We've seen carbonic anhydrase a lot throughout our course here. And so we're going to now finally understand what the enzyme active site looks like and how it does its chemistry. And it also uses water, right? We know that's what carbonic anhydrase does. Water plus carbon dioxide makes carbonic acid. Okay, so let's review here with our polymers. And I'm just going to kind of go through them one at a time. We'll highlight the name of the enzyme that we're going to be studying. So carbohydrates have glycosidic linkages. And we are going to study the enzyme lysozyme. So lysozyme is a enzyme that we make that helps weaken bacterial cell walls. And so we'll get to the details of that when we study lysozyme. Then we've got nucleic acids. Specifically, we're going to talk about RNA. And so we've got our phosphodiester bond. So our phosphodiester bond, and we're going to specifically be studying RNase A. And I should have had, that's a lowercase a, RNase A. Sorry about that, uppercase A. And then we are going to study proteins.
One sec.
Oh, this is going to be a problem when we study our proteins. You'll see why in a second here.
Working on that.
When we think about proteins, we've got peptide or amide bonds. And we are going to be studying serine proteases. That's usually a problem when I say that. All right, so those are the 4 enzymes that we're going to study: lysozyme, RNase A, the class of enzymes, and we'll talk about why it's a class of enzymes, serine proteases, and then we'll also learn about carbonic anhydrase. Okay, so we're going to see some general themes for how amino acids can help do chemistry. Okay, so we're going to highlight what those are right now, and we're going to highlight that there's really maybe a subset of about 8 amino acids that you really need to know, even though you have to know all 20 of your amino acids. Remember, many of them are those nonpolar amino acids that just create that hydrophobic core that stabilizes protein tertiary structure. The ones that have the weird letters, those are going to be the ones that have functional groups that are going to allow us to do some unique chemistries. So let's talk about what those chemistries are. So one of the chemistries that we're going to see is the addition or the removal of protons. Remember all of those reaction mechanisms that I had you do, right? And I told you we can't just write PT for proton transfer. I want you to show if you're adding or removing a proton. And here's why, because those are going to show us places where we can have amino acids help do those chemistries in enzyme active sites. And so when we think about what amino acids might be able to help with that, there's really only 5 that have reasonable pKa values. So there's 5 with reasonable pKa values. Okay, and so we're going to think of these as being our acidic residues, which are negatively charged, and then our basic residues, which are positively charged. And I know sometimes this can be confusing because you're like, basic residues, bases, those are things like hydroxide that are negatively charged. Nope. When we think about acidic amino acids, those are our glutamic acid and our aspartic acid, right? And because they are acidic, they lose their protons and they become negatively charged. So in this case, okay, we're going to have ones that have lost their protons. So they're going to be ones that can actually often help by taking back protons, okay? So then we've got our base. So let me write down, these are our acidic amino acids. And let me actually change, let's do this so just so we can keep consistent with things that we sort of see when we see these amino acids mapped onto protein structures. These are going to be spaces where we see red. So let's put these in red for our acidic amino acids. So glutamic acid, aspartic acid, they're going to be negatively charged because they've lost their proton, but that means that they are going to have the ability to grab back protons, okay? Or as we're going to see in one enzyme active site, we actually do have a glutamic acid residue that has a pKa that's shifted to a higher value, so it retains its proton. We'll get to that one later. Okay, so in blue here, I'm going to put our basic amino acids. Okay, so it's going to be our histidine, our lysine, and our arginine. So histidine, lysine, arginine, they are positively charged, and that's because they have a proton. So when we think about how these amino acids can help, these are going to be ones that can add or remove protons. But again, that depends on what their starting protonation state is. So again, we'll see typical situations where they sort of start with what we'd expect their protonation state to be, but then we'll also see an example where we have pKa shifting. And then we're going to have a special amino acid, histidine, that has a pKa near neutrality. It's just a little bit above 6, but that's important because that actually allows it to have both capabilities of adding or removing protons. I want to make sure to highlight here that many of the amino acid residues we are going to see here are the ones that have those strange letters. And what do I mean by strange letters? The letter that doesn't match the starting letter of that amino acid. But remember, when we learned about why those letters were assigned, those single letter codes were assigned the way that they were, the reason is because the amino acid that has, like, take alanine, that's more prevalent than aspartic acid or arginine. And so it gets the A. So the ones that we have here are generally not going to be the first letter of that amino acid. And that's because they are found more rare because they have unique functional groups that really allow them to do unique chemistries. Okay, so adding and removing protons is going to be a very common thing we'll see amino acid residues do in our enzyme active site. Another thing that they can do is they can help with charge stabilization. Remember, when we learned about how enzymes do their chemistry and we looked at reaction coordinates, we said that they often bind to and stabilize the transition state. So if we're going to be binding to and stabilizing transition state, one of the ways that we can do so is by stabilizing charge that's developing on the transition state. So again, things that we're going to see are these charged residues. We're going to see our histidine, our lysine, and our arginine. Okay, these being positively charged are going to help to stabilize things that are negatively charged. And then when we have our glutamic acid and our aspartic acid, these are going to be negatively charged, so they're going to help stabilize something that's positively charged. And highlighting here, when we're talking about doing this stabilization, I just mentioned it, but just to make sure to highlight it here, why this is important, because this is often what decreases the transition state energy. And that's how enzymes work. Enzymes work by stabilizing the transition state, making for a smaller delta G double dagger, which makes a spontaneous reaction go faster. Okay, last thing here to highlight is a common way that we can see amino acids facilitate chemistry is they can act as a nucleophile. Remember, there's amino acids that have oxygen and nitrogen atoms as part of their R-chain functional groups. Those are ones that are going to be able to participate in chemistry as a nucleophile. But often what we're going to see here is we're going to see amino acid residues that act as water surrogates. So we're going to see often, and this is important here when we're talking about these hydrolysis reactions, they're often what we call water surrogates. They're acting in the place of water. And so when we think about amino acid residues that have the ability to do that, we've got serine, threonine, and tyrosine. And specifically to highlight here, this is the namesake for why we are learning about these guys. So we've got serine proteases, okay? And then the last thing to highlight here is when we think about things that we still need enzymes to do that maybe amino acids can't help with, one of the things that we're going to see a lot is redox chemistry. Remember our important fuel equation. Fuel plus oxygen goes to carbon dioxide, water, and energy. That's oxidation of our fuel. We're gathering and harnessing those electrons. These are things that amino acids cannot help with. So this is not helped by amino acids. They don't have the ability to participate in redox chemistry. So that's why we're going to see things that we're going to call coenzymes. We already have learned two of them, right? We've got NADH and then FADH2, or found in their oxidized states, NAD+, and FAD. But highlighting, and we already know this, and you're going to have a vitamins quiz coming up soon, right? NADH is really vitamin B3. FADH2 is really vitamin B2. But one of the things that we are going to see is that our B vitamins are going to play critical roles in many of these coenzyme activities that we're going to see in understanding how enzymes work.